Okay, so let's get started. Welcome to the first seminar of the Theoretical and Computational Biophysics group in the fall 2021. So I'm very pleased to host Dr. Amy Shih, actually a former group member from TCBG and from Beckman Institute, to start our seminar series for the fall. Before I introduce Amy, again, I remind everybody to please turn off your microphones to avoid any background noises to interfere with the seminar. And if you feel comfortable, turn on your camera so we can see you and your face and your office and your nice backgrounds if you want. And then if you have any questions, I'm going to invite you at the end of the seminar to ask your questions yourselves, so to have a more interactive experience with the speaker. So, and with that, so let me introduce Dr. Shih. So Amy is a native of Michigan, where she did her initial training in biochemistry, University of Michigan, Ann Arbor, and then she got a master's degree in computer science. I think that's how she got attracted or gravitating towards computational sciences from Michigan. And then she moved to Illinois, where she joined the biophysics and computational biology program. It used to be called biophysics and computational biology. PhD program in Illinois, University of Illinois, Urbana-Champaign. And she did work with late Klaus Schulten and Steve Sliger on her thesis, concentrating on lipoproteins, experiments, and modeling and spectroscopy. In 2008, she received her PhD in biophysics, and then immediately after that, she received actually a prestigious postdoctoral fellowship from Beckman Institute for 3 years, during which she stayed with Klaus and did additional work on lipoproteins and also P450 system during that period. So in 2011, actually, she made a decision to move to industry, and she joined Janssen Research and Development as a computational chemist, initially as a postdoc. And after some transition to other companies and back to Janssen, she was actually promoted to a senior scientist and then later principal scientist in 2019. And just recently in 2021, she moved to another company, if I'm not mistaken. Ferring Pharmaceuticals is the name of the company, and she has been the associate director and head of computational chemistry in Ferring Pharmaceuticals since earlier this year. So as you can see, so with very nice experience in membranes and membrane proteins, membrane associated proteins, she actually moves to pharmaceutical setting and starts to apply these skills and computational tools to real world problems, namely drug discovery and understanding receptors and drug targets and so on and so forth. So I think, as you can see from the title of her presentation, which is in the Zoom already, so Amy is a perfect person to combine what you guys are doing in academia and what is happening in industry. And when we invited her to give us a seminar, so we were hoping to have a kind of combined academic and career-oriented presentation by her. And I hope that she doesn't mind to be asked a lot of questions at the end in case any of you are interested in details about industry and how it works in the industry and how to join industry and so on and so forth. Well, thank you very much, Amy, for accepting our invitation and for being with us and for being back, because I wished we could have you here actually and have you physically in person, and maybe we can do it at a later time, another time. But for now, so thank you very much, and we look forward to your presentation. Please.
Great, thanks, Ahmad, and thanks for inviting me to present today. I can't tell you the number of TCBG seminars I've attended over the years, but as Ahmad mentioned, this will probably be very different than your typical TCBG seminars with very little sort of scientific depth and really just more focused on my path to industry, and then sort of, what computational chemists do in industry, what types of methodologies are used and things like that. And we can certainly discuss, paths to industry, what methodologies and things like that are used in industry and all those types of activities as well. So hopefully I budgeted enough room at the end for a Q&A, but I can also stick around for additional time if there are additional questions. So my path to industry, and I put serendipitous in capital letters, because honestly, if you asked me when I was a child or even in undergrad, pharmaceutical industry was not really on my radar at all. For much of my childhood, I wanted to be a medical doctor, a real doctor, and it really wasn't until sort of halfway through my undergraduate career that I really thought about science as a career as opposed to medical. And so, I find it very ironic when I get asked questions like, you seem to have a really well mapped out career path that, how did you know you wanted to go into industry, or how did you know, how did you know you want to go into pharma? I really didn't. I actually didn't consider pharma and really major or large pharma until probably the end of my graduate school career. People ask me all the time, how did I know I wanted to be a computational chemist? I didn't. Honestly, even when I went to industry, I didn't really understand what computational chemists did in industry. And so, I say very serendipitous, because really my career path was based off of my interest at any given time. And sometimes, what drove me to on one path actually were not necessarily great reasons for choosing particular paths. So, probably most people here who know me from the Beckman Institute, where I did my PhD. But really for me, my path to industry really started before that. And really it started, I would say, in my Michigan years at University of Michigan, where I did my undergraduate degree in biochemistry. And really, I was purely a experimentalist at that time. I did biochemistry experiments in my junior year of undergrad. I actually joined a lab in the University of Michigan Medical School in the allergy and immunology department, which was actually focused on antimicrobial emulsions. And so I actually worked there as a lab tech. So, the person making, the lowly student making lab reagents and pouring Petri dishes and things like that. But they actually slowly got me integrated in doing some of the antimicrobial research that the lab was doing. And really by the time I graduated, I was fully involved in doing the academic research that was being done in that lab. And so they offered me a position when I graduated, and so I joined as a research assistant in Dr. James Baker's lab. And really, this is where I got my first exposure to academic research and research in general. And I really think this is probably where I learned how to do research in general. It's really where I got my first publications in scientific publications, as well as my first patents. And they were all around antimicrobial emulsions. So after I had been at the University of Michigan Medical School for about 2 years, the technology had advanced to a point where they decided to spin it off into a startup company. So I became employee number 3 of NanoBio. It was I'll call it tiny biotech company. Even when I left 2 years later, it had only about 10 employees. And that, I guess, gave me the first glimpse of what industry research is. I put industry in quotes because, any small startup biotech that originates from academia, it's still pretty academic in nature. So I will say, I call it industry research in that I did get exposure to, things related to business decisions and things like that. But really, it was the research part of things was still fairly academic. At NanoBio, I did get the opportunity to do a lot of things because of the size of the company that I really probably would not have done anywhere else, and that included setting up an experimental laboratory, writing SOPs for GMP manufacturing of late stage clinical material and things like that. And so lots of exposure to things that a normal scientist would not necessarily do. But I really enjoyed that. It was probably the most fun I had in any career. And that was really what I enjoyed. Now, during this time, I did I would call it my spare time. I did a master's in computer science at the Eastern Michigan University. And and really I did this at night, mainly because I was interested in computers and how they could, and computational methods and how they could be used as tools in scientific research. I dabbled a bit in my undergraduate career in sort of a few programming classes, but I really wanted to at least take some more and really understand how computational methods could be used in biological research. And I also got both the University of Michigan Medical School and NanoBio to pay for it, so that was also a bonus. So I will say, I finally finished my master's degree in 2003. And really all during this time, my PI had essentially been pushing me to go to graduate school. And I always had in the back of my mind that I would go and pursue a PhD at some point. And so really that was where, when I finished my master's, I finally decided, okay, I will go and do a PhD. I had a lot of options at that point, including staying at Michigan. My PI happened to be the head of a nanoengineering department at Michigan, and I could have easily stayed and essentially continued the nanoemulsion research that I had been doing for the last 4 years. But I really wanted to go somewhere where I could also combine the computational aspect of the research that I was interested in. And so that really is what drove me to University of Illinois, and the biophysics and computational biology group. It's University of Illinois has a very strong computer science and computer engineering department, and I really thought that would be a good area where I could really find a project that could combine both biology and computational methods. And so I'm thinking you guys are doing this right now, according to what Imad said. So, early on in your when you first go into grad school, you get these seminars by PIs who are looking for graduate students. And so I went to these seminars and I picked my 3 rotations based off of them. My 3rd rotation, I picked Stephen Sliger. And really it was because of his interest in the research that he was doing on the nanodisc system. The sort of nanotechnology aspect of things, which I'd sort of been working on essentially, for most of my career, although from an experimental standpoint, was really drew me to the nanodisc system. Well, there's one other thing that really drew me to his lab, and actually this is probably the deciding factor for me to going and doing my rotation in his lab. He put up a picture of Bar Harbor. Now, I actually don't remember why he put up a picture of Bar Harbor, but I love Bar Harbor. It was my favorite place to go on vacation. And so when I saw that picture, I wanted to do a rotation in his lab.
I should include a nice picture in my presentation tomorrow.
The things that draw you, I really don't. I'm not suggesting that a picture of Bar Harbor should be anyone's decision factor for joining a lab, but that really was one of the reasons I chose Steve's lab to rotate in.
Very interesting.
And so, when I went to his lab and we were discussing sort of projects that I could do, as a rotation student, he looked at my background, saw that I had a computer science background. And so the next thing I knew, he was asking me if I could do modeling on nanodiscs. And I was like, sure, why not? And then I ended up essentially, for most of my rotation, in Klaus's lab instead. So, and so really that's how I ended up with sort of joint advisors, one experimentalist, one computational, for my PhD career. And so, as Imad mentioned, I essentially focused on modeling and self-assembly of nanodiscs for my PhD career. And Steve's lab, I obviously got a lot of rigorous training on small angle X-ray scattering, which is a biophysical technique looking at sort of the shape and size of compound proteins and lipids. And then obviously in Klaus's group, I did a lot of molecular dynamics and obviously a lot of coarse-grained molecular dynamics that I was really developing to enable sort of the simulations that I wanted to correlate with the small angle X-ray scattering experiments that I was doing in Steve's lab. And so, let's see. So this is actually a picture of the Argonne National Lab beamline that we actually ran our SAX experiments on. The sample sits right about there. And actually, funny enough, my favorite memory of the Argonne National Lab is riding tricycles around, trying to keep awake after 72 hours being at the beamline. So that was probably my favorite thing to do. In Klaus's lab, obviously the rigorous training we had in TCVG, but really the exposure we had to high performance computing is something that has actually stayed with me and actually is very useful in computational chemistry groups in industry, because obviously we're very dependent on high performance computing. But despite all the rigorous or scientific training in biophysics and statistical mechanics and molecular dynamics, I would say probably the most useful thing that I learned at UIUC and really during my graduate school career is communication skills. And when I say communication skills, I don't mean communicating with other computational chemists, computational biologists, theorists. It's really the skill to communicate with non-computational, non-theoretical people. And really, I credit that to, number one, to Steve's group. That I can't tell you the number of group meetings that I presented at, but it was really a learning experience for me to be able to communicate with a group that was completely full of experimentalists. None of them had any molecular dynamics background or modeling background. And so for me to communicate what I was doing in Klaus's group to them, it was a learning process for me. And definitely that skill has carried over. The second aspect where I really learned sort of communication skills is, and I don't know if that room is still the same room, but that room used to be a 3D room when I was at TCBG. And Klaus used to have these 3D room demos that he made us all prepare. They were supposed to be 5 to 10 minutes long, very sort of visually appealing, quick descriptions of your project and sort of the key discoveries and things like that, and really distilled down in a way that was very easily understandable. And really the audience was meant to be very, very diverse. So anywhere from your professors who are visiting for your TCBG seminars, all the way down to elementary school kids who are visiting for Beckman open houses. And that really helped in sort of thinking about how to make things visual and making things in a way that a very diverse audience can understand. And I say this because computational chemists in industry work, when they're doing project support, they work usually as the only computational chemist in a drug discovery group. And everyone else in that group is not a computational chemist. And so you really need to be able to communicate your results in a way that other people will understand and really understand how to utilize your results and really and the impact of your results on the drug discovery project. That is really a skill that I feel is by far the most important and one of the most important things you'll have going into industry. Okay, so I graduated in 2008. As I was finishing up my PhD, I really started to think about what I wanted to do as a next step in my career. And I thought I knew what industry was based off of my experience at NanoBio. I can say that probably was not the case, but I really liked industry and I liked pharma. I liked the idea of really applied scientific, where really you were hands-on in discovering drugs that could eventually benefit patients. However, if you can remember, 2008 was not exactly the greatest time to be looking for a position, specifically, definitely not in industry. Most of industry was essentially either not hiring or essentially laying off, and actually laying off for many years. And so I don't know if anyone knew this, but the Beckman Institute Fellowship was actually my backup plan. I thought I could do it for a few years and make sure that I really focused on targets that would be of pharmaceutical relevance. And really that's why I focused on things like cytochrome P450, which is an enzyme that's of particular interest to the pharmaceutical industry because it metabolizes a large majority of your small molecule drugs. And so obviously during my PhD, I worked on cytochrome P450 and obviously the nanodisc system. Now, 2 years later, when I was finishing up my postdoc, I again thought about pharma. It still was not a great time to be finding a job in pharma. And I actually had a backup plan. I don't know if anyone knew this, but I actually became a registered patent agent during my postdoc fellowship at UIUC. And I was actually exploring becoming either a patent agent in a law firm or going to a university tech transfer office. I actually had multiple offers. But instead, just before Thanksgiving, I got a phone call from Janssen in San Diego asking me if I wanted to go and interview for a postdoc fellowship there. And it was literally, something I had applied for almost 9 months ago. And so I'd already given up hope on this. And so I was like, I don't know. I don't really know if I want to do another postdoc. I don't really know if I want to go to California, but I figured I at least owed it myself to go and see what, the group was like and things like that. And so I flew out just after Thanksgiving. I interviewed, and I really loved the group there. I loved the hiring manager. It was really a fantastic group, a fantastic environment. And so the next day, when I was sitting on the beach, just about to go for a hike, they called me and asked me if and offered me the position. And I thought, well, okay, it's California, but I guess I can deal with the sun, because literally this was what my backyard looked like the day I left for San Diego. And I thought, okay, well, at least I can handle this for a few years. And so that's really how I ended up in San Diego, and still in San Diego and at Janssen to do my postdoc fellowship. So Janssen, perhaps better known as the pharmaceutical wing of Johnson & Johnson, and probably perhaps best known these days for its COVID vaccine, is most definitely large pharma. You don't get much larger than J&J. Janssen is the pharmaceutical wing, but J&J also has a consumer products division as well as a medical device division. And at Janssen, Janssen has 3 major research sites. One in San Diego, which actually is the smallest, another in Springhouse in Pennsylvania, and the third is in Belgium in Beerse. The San Diego site was about 350, 400 people. And it's primarily, it's solely essentially a research site. There's almost no clinical activity or developmental activity at the San Diego site. And its primary therapy areas of focus were neuroscience and immunology. And so during my postdoc, Janssen had a really sort of strong philosophy that it wanted its postdocs to work on projects that were publishable in I want to say a postdoc lifespan, so in a 2-year lifespan. And that's very important, because a lot of if you work on an ongoing active research project, you may not be able to publish on that in a 2 or 3 year time span. And it's very important to publish, because that's what you're going to go to when you interview. If you do not publish, you will not be able to present on anything you do in an industrial postdoc. And so that really was a focus of Janssen. And so I worked on a lot of projects with other postdocs that had sort of been predefined as things that could be fairly rapidly wrapped up. And so, I had a number of publications on GPCRs and modeling of GPCRs, as well as some on sodium ion channels and this spider toxin. It's actually a bird-eating spider toxin. If anyone's ever interested and really enjoys bird-eating spiders, there's a lot of YouTube videos out there on it. So not exactly what I wanted to work on, but that's what I ended up working on. So, but despite all the sort of the publications and the projects I worked on during my time as a postdoc, I would really say that it was sort of the things that I got exposed to, that I got, introduced to, that had nothing to do with the projects, but were really more, that really benefited me in finding my next job and really understanding what computational chemistry did in industry. And that's, I got exposure to computational chemistry and how they operated within drug discovery groups. So, I got to shadow some of our more senior computational chemists. And I actually got to work on some of their projects. And I was telling Ahmad earlier, I actually ran MD on a project that another computational chemist was supporting during my postdoc career. And later on, when I went back to Janssen, and almost 8 years later, that actually became my project. So some of the longevity of some of these projects can be quite significant. So besides our exposure to computational chemistry, you actually get exposed to all the other functionalities that are involved in drug discovery. So that can be your screening groups, your therapy areas, it can be your structural biology groups, it can be your DMPK colleagues, it can be your compound profiling group, it can be your cheminformatics or compound logistics groups, computational biology groups. All these groups and functions you get exposed to on a daily basis in pharma. At Janssen, you also had the added benefit of not only being exposed to sort of clinical and business and manufacturing sides of sort of pharma, we also got actually they had a strong sort of cross-sector initiative where they actually got people a lot of exposure to sort of the other business divisions, so the medical device and consumer products areas as well. You get a lot of exposure to external vendors. So, your software and hardware vendors, you get to meet all of them, and you get to, and later on when you're evaluating software and things like that, these are probably the people you are interacting with, and they do stay with you for a long time. So, the sales rep I had for Schrodinger in my postdoc days became my sales rep even now. So, so things like that. And networking with people in pharma, both within, Janssen, but also in other companies, it's invaluable. Those are the people you will be reaching out to for the rest of your career, in all aspects, whether it's to discuss ideas or to find your next job. Okay, so after Janssen, I did my postdoc for about a year and a half. I got a position at Dart Neuroscience. Dart Neuroscience is what I call a medium biotech company. It was about 250 people, privately held company that was solely located in San Diego. And its main focus was on research on drugs to enhance long-term memory. And for a company Dart's size, they actually had a fairly sizable computational chemistry group. So it both had a computational chemistry group that was dedicated to supporting their drug discovery projects, which I was a part of. They also had a computational chemistry group that was actually involved in methods development, which is actually very rare. And Dart was actually a really fun place to work for as a computational chemist, probably for one reason, well, maybe multiple reasons, but one of the main reasons is it actually had a medicinal chemist that headed the medicinal chemistry effort that was very pro-CAD, very pro-computational chemistry methodology. And that sort of thinking permeated all the drug discovery projects. And so computational chemistry ideas and predictions and calculations was actually highly valued. I can tell you that is not always the case, especially in large pharma. You will encounter chemists who don't care a single thing about computational chemistry and just want to do their own thing. So, working with people and things like that, and even people who are resistant to your ideas are part of something you're going to have to do if you're in large pharma for sure. That I didn't need to do that at Dart because of the sort of the environment that we're in. Actually, one of my favorite stories I can tell of my time at Dart is this, I'll call it a beer bet that I had with the head of the medicinal chemistry group on the binding pose of one of my targets I was working on. He had actually worked on that target previously at another company, and he had lots of experience with this tool compound that we were doing some analoging off of. And he had a very set in his mind that compound bound in a very specific way in the binding site. And I docked it, and did MD on it and docked it some more. I told him, no, I think it stocks exactly 180 degrees. And we literally bet on it. And a year later, the structural biology group was actually able to get a crystal structure of the GPCR, and in fact I was right, so I won my beer bet. So fun, exciting times there. Now, how did I end up back at Janssen? So 2 years after I joined Dart, out of the blue, I got a phone call from my manager at Janssen, my former manager at Janssen, asking me if I knew that they had an open position, which I did. And she asked me, was I going to apply for it? I'm like, no, you're looking for a senior scientist, somebody with like 5+ years of experience. I only have like 2. I'm happy at Dart. Long story short, she convinced me to apply, and obviously I ended up back at Janssen. To be fair, that was a group I really enjoyed, and Janssen actually has a lot more diverse targets than Dart. Dart was actually very narrowly focused, and I really wanted that at an early stage in my career where I could really work on a large number of targets. And so at Janssen, I had the opportunity to work on a lot of different therapy areas, anywhere from immunology, neuroscience, oncology, and infectious disease. I worked on a lot of protein-protein interaction targets, a lot of GPCRs, a lot of functional proteins with a lot of dynamic movement and conformational changes. I got to work on projects that had small molecule leads that came from all kinds of sort of lead finding strategies. So including things like, projects where literally we're just taking competitor compounds and running a fast follower program. So trying to get around their IP to develop a drug, all the way to, fragment-based methods, your traditional high throughput screening methods, or even things like DNA encoded library, where you're really looking at screening much more billions of compounds, and virtual screening methods. So I actually got to work on lots and lots of sort of different lead material during my time at Janssen. At Janssen, I was, given sort of increasing opportunities for leadership. I really, probably one of the things I started even back in my postdoc days was really champion the use of molecular dynamics in some of our projects, especially projects where there were known conformational changes in the protein that resulted from activation and things like that. And I ended up chairing the the molecular dynamics and free energy working group, and we're as telling Ahmad, yes, we did run a lot of free energy calculations. We evaluated a lot of free energy methodologies, which I'll actually talk about a little bit later in this meeting. I also had the opportunity to have postdocs of my own during my time at Janssen and things like that. So I was also heavily involved in things like ultra high throughput screening and ligatability and things like that. I actually had a number of my projects go to clinical candidate nomination. And actually, one of them went first in human earlier this year. So, as a computational chemist, and really anyone in early drug discovery, that's always your goal, is to get sort of things into clinic, obviously, eventually out to market, but first steps out to clinic first. So earlier this year, I decided I would join Ferring Pharmaceuticals, and where I am now associate director and head of computational chemistry. And really at Ferring, I was really recruited to head the group because it was really a group that was being newly formed, and so really I was being recruited to form the group and really define how computational chemistry was being used at Ferring. So very quickly, what is Ferring? So Ferring, if you know Ferring, it's probably because of their in vitro fertilization drugs. It's a privately held company that has essentially is a leadership position in reproductive health and maternal health, and some specialty immunology and particular gastroenterology therapy areas. It is a global company with sort of research facilities, or sorry, development facility and sort of manufacturing and things like that globally, but research-wise, it's primarily in San Diego and in Copenhagen, Denmark. And I'm showing this slide not because I want you to, or I'm going to talk about any of these medications, but really I'm showing this because for 70 years, or just shy of 70 years, Ferring was really primarily a peptide-focused company. It was founded as a peptide company, and essentially most of its drugs are peptidic drugs. A few years ago, they decided that they no longer wanted to be just a peptide company. They wanted to be, let's say, modality agnostic. So they wanted to incorporate both small molecules and antibodies or biologics. And really, rather than focusing on peptides, they wanted to focus on the therapy areas of particular strength to the company. So that's reproductive health, and maternal health, and immunology. And there was also the realization that, with the shift from peptides to other modalities, that there were functions that did not exist at Ferring that really needed to exist to enable those types of efforts. And so computational chemistry is really one of those functions that was decided that, they really needed to bring in to support both small molecule and biologics programs. Other functionalities were also brought in, things like high throughput screening, structural biology, and things like that. So all those are essentially got started, let's say, earlier this year. And so computational chemistry at Ferring is really meant to be applying cutting edge computational methods to support our drug discovery portfolio. And that means any of the portfolio, small molecule, peptides, or biologics. And so, much of my early, 8 months that I've been at Ferring has been in really setting up and building the group. So, Sunny was our most recent hire, but we are now a group of 3 computational chemists. We also have a number of external computational chemistry consultants and IT consultants who also help with our projects and things like that. A lot of setup of hardware and software infrastructure. So, going back to high performance computing, a lot of companies no longer are setting up internal clusters, but are relying on cloud computing resources and things like that. And so those have been things I've also been working on, is setting up cloud computing resources to augment our sort of internal workstations that we have. And then obviously we have a mixture of sort of commercial software and free or free software. So outside of sort of just computational chemistry, there was also the need for sort of a more drug discovery centric data visualization informatics platform. And so obviously computational chemistry is also really involved in setting up that platform, making sure all the data gets in there in a proper way that we can essentially extract it for data analytics and things like that. And we're also deploying molecular modeling and data analysis tools to project teams to enable them to do their own modeling. We're also heavily involved in setting up and evaluating compound libraries. So, keep in mind, small molecule compound libraries for things like HTS screening, fragment screening, and things like that are very important for lead generation in pharma. And so we are actively involved in sort of identifying compounds of interest for purchase. We're also heavily involved in sort of external innovation opportunities. So this can be collaborating with not only small companies or biotechs or things like that as they bring on new technologies, but also academic groups as well. So, okay, drug discovery funnel. How much time do I have? Okay, so I'm going to go over this really quickly. So, what is drug discovery? And I'm thinking most of people have seen this. The sort of the narrowing down of compounds to your final clinical phase compounds. So generally at the early discovery stage, you generally have thousands, I would say this is actually probably billions, but so you're looking at billions of compounds, and then you're essentially going and optimizing your compound, your drug into a 1 or 2 lead series that eventually get into clinic. And so, one of the questions I get asked a lot is, what, no, that's not the question I asked. This is the question I was asked. What parts of drug discovery are computational chemists involved in? And so really, computational chemistry is involved in all early discovery and as well as even into some of the preclinical development phases of things. For the most part, once a compound is identified, there is not a lot of research. Well, there is research done, but generally research in small molecules, at least in this sort of this, winter went away, in this development area are around formulations. So generally you're looking at sort of identifying and optimizing your drug in this range, and then looking at formulations and really how you deliver the drug in this stage. And for, let's say, the sort of the development area, depending on the company, not all companies have computational chemistry efforts in this area. There are some companies that actually have significant efforts in this area, and especially in small molecules, Merck and things like that actually have significant preclinical computational chemistry groups that do crystal form predictions and things like that. Another sort of commonly used, or let's say increasingly commonly used area for preclinical development is actually in encapsulation, formulation, liposomal encapsulation of drugs and things like that. I don't know how well known it is, but like companies like Moderna and their mRNA vaccines, they actually had an entire computational chemistry group which was looking at encapsulation of their mRNA vaccines, and actually they ran tons of coarse grain modeling. I don't know the results of that, but that was a particular effort of theirs in the development phase. But for most of this, I'm going to talk about essentially the discovery phase of things, because that's the area that I'm most involved in. Okay, so what do computational chemists do in pharma? So we are members of drug discovery projects. So literally from target selection all the way to clinical candidate selection, we are core members of project teams. We're heavily involved in informatics infrastructure, so the data analytics, things like that, data visualization, and methods development. I would say, at least in large pharma, methods development is, I would say, increasingly less found, decreasingly found. Yeah, decreasingly found. Methods development really is more in biotech and in software companies these days, and large pharma generally will collaborate with biotechs and small and software companies to do the method development. There is not significant method development being done in large pharma anymore. Okay, messed this. Okay, we'll go to this. So, and this is a question I get asked probably the most is, what methodologies are used in pharma? And the only way I can respond to that is lots. Essentially, we're looking at anything from target selection all the way down to clinical candidate selection. And what methodologies you use at any particular point, it can be very different depending on the stage of the project and what the needs of the project are. And really, one of my former managers, used to call computational chemists in industry a jack of all trades, master of none. I would argue that we're actually master of one, and generally this is what you did in your PhD career. That's the one you master. Everything else is learned. And so really that is computational chemistry, especially the project support computational chemists. Okay, so now I'm going to talk about some of the methodologies that are used in computational chemistry in industry. This is by no means a comprehensive list or even a representative list. This is really just what I thought about over the weekend as I was putting together the slide deck. So I'll just give you that heads up. Liganability. So liganability analysis early on in target selection is actually quite important. So, if you have a structure, trying to determine whether there is a pocket that's amenable for small molecule drug discovery is important to de-risking your target. For a target such as a kinase with a known small molecule binding site, things like that, it's not really a huge hurdle to sort of do a liganability analysis. But for targets such as protein-protein interactions, where you generally have a very large surface where proteins are interacting, trying to see if there's any sort of amenable pocket in that interface region or in a surrounding region which may be amenable to drugs, small molecule drugs, is actually very important. And then obviously if there's targets where there are no known functionality and things like that, trying to predict whether there is a small molecule binding site could be important. Historically, there's been sort of these static structure grid-based methods, and these are fairly old. One very common method is using chemical fragment probes, so like a phenylalanine and things like that, benzoacetamide and things like that. Boston University, Sandor Vajda has this FT-map method that is widely used, which uses these chemical probes over a grid and essentially minimizes over the grid to sort of give you an idea of potential hotspots. And it's a fairly quick method. He actually has a website that you can submit things to, or you can obviously in pharma we buy licenses to the software and things like that. Schrodinger and CCG have, I call it the site points alpha sphere. I call them dummy probes, but essentially they have spheres that are again used to probe the surface of a protein using a grid method. And really they're calculating essentially hydrophobic propensity, concavity, as well as potential polar or charge interactions. And really they're using that to sort of determine whether something is a good binding site and to give it a ligandability score. For these static structure methods, we actually often use all of them just to get a consensus. There's not one that really necessarily does better than the other, at least in my experience. So we tend to do all of them. Now, the problem with these is obviously they rely on static structure. If there's any cryptic pockets or allosteric pockets that open up, none of those will be found using these methods. And so really that's where these sort of dynamic structural methods that have come into play. So things like Pele, which is now being commercialized by Nostrum Biodiscovery, where they use a Monte Carlo method to really allow sidechain movements and even small backbone movements, and trying to really allow for cryptic pockets and things like that to open up. Actually, this is a example from Janssen, where they had a flu virus hemagglutinin, and actually the compound actually binds in this area, but this area is actually in the apocrystal structure is actually not it's a closed binding site. So this compound would not be able to dock in this area. in the apo structure. And so they actually did a retrospective analysis where they took that apo structure, ran the Monte Carlo based Pele method, and actually got the compound to bind in that method, in that binding site. And we actually had the comparative result because we actually had the bound structure, and literally that the predicted structure bound in the exact same way that the crystal structure bound. I'll say this is not necessarily representative of the success of these types of methods. We've run these on a lot of other targets, and so sometimes they work, sometimes they don't. There's a lot of sort of MD-based methodologies. So Silks is one common one that's out of McCarroll's lab, using, I think it's Glomax based, as well as coupled with the Monte Carlo moves. So both of these, mixed solvent MD out of the Carlson's lab that's now in Schrodinger. So both of these methods essentially use chemical probes coupled with molecular dynamics to essentially try to interrogate binding sites and really map, where potential interactions could be beneficial to free energy. And so, these are all methods that I've actually played around with in the past, and we often will use these in conjunction with the static methods to try to look at ligandoability. Cheminformatics. So I'm guessing this is an area that probably very little of people from TCBG backgrounds know very much about. So cheminformatics is essentially using compound data and coupling it with any other assay data you have, compound related data you may have, DMPK related data you may have, etc., and really using that to help drive your research projects. And so a lot of this is based off of the corporate compound collections that we have access to. So obviously, there's a lot of data in the public domain. I say the pharmaceutical industry's sort of compound databases, the related and associated data are by far the largest wealth of data, and sort of that can be used for really enabling drug discovery projects. And the analysis of that data is really something that most pharma does a lot and spends a lot of time doing. So, we spend a lot of time annotating our library and then analyzing our data, our library as well. So this can be anywhere from identifying frequent hitters that show up in our high throughput screening assays, things that are technology hitters. So they could be like a fluorophore or something like that. Compounds that are known to be aggregators that can sort of skew our assay results, or compounds that are known to be insoluble or have ADME property issues, CYP issues, and things like that. And really these compound databases and the associated data are really the basis for a lot of the QSR and machine learning modeling that we do in pharma. And we do oftentimes incorporate public data sets. Let's say the quality of the public data sets generally is not of the, a lot of the data in public data sets are, I'll say, a little bit questionable. And so generally the pharmaceutical companies' data sets are much more reliable and predictable in use in generating models. Obviously, compound libraries are also of use for things like HTS high throughput screening assays, as well as virtual screening when we're doing lead generation activities. Typical sources of compound collections are things like, internal projects, external vendor compounds, and then historically there used to be combinatorial chemistry efforts, although those are now primarily in the DNA encoded library and virtual library space. So very few pharma companies are now doing combinatorial chemistry efforts. Just give you an idea of sort of, how elaborate sort of pharmaceutical companies go to enriching their compound collections, outside of looking at things like diversity, or, if there's a particular target class of interest, and really biasing our collections to that bias, to that target class. At Janssen, we actually set up a voting system for our chemists to actually vote on whether they liked a compound or not, and would be willing to work on a compound if it was found as a hit in a drug discovery project. And it was actually kind of interesting because it varied a lot by chemist and even by site. So there were definitely strong preferences that really sort of biased what tolerance people had to to starting material in drug discovery campaigns. We actually had a postdoc where we actually went back and analyzed the performance of our HTS library and really trying to see, how useful it was in terms of generating hits and where the hits came from. And actually, you can see that essentially most of the hits this was actually a HTS analysis done over almost 80 HTS campaigns over 5 years. Most of our hits actually came from internal project sources. And a third of them came from external library purchases or design libraries. Virtual screening. So this is sort of a complementary type activity to some of the activities that the various experimental screening groups do. So these are really ways that we as computational chemists can accelerate lead identification. And so there are definitely, your ligand-based virtual screening methodology. So these tend to be much faster. So 2D and 3D searching options. Docking is something we do routinely. Machine learning-based algorithms are actually becoming more and more used, sometimes in conjunction with your ligand and structure-based screening methods, sometimes on their own. So we'll have to see how they perform over the years. And when I say virtual screening for lead identification, we can screen things from anywhere from our internal HTS collection or our entire corporate collection. We can screen external vendor libraries, but probably the most sort of impactful thing that we can screen are virtual libraries. And these are libraries that are highly curated to be synthetically accessible. So they're built off of building blocks. And the synthesis of these compounds based off these building blocks is fairly well known. So it's sort of fairly vetted to be able to synthesize these compounds. And there are vendors that have these types of compounds, and a lot of companies have their own virtual libraries. And so just to give you an idea of sort of the scale we're talking about. So your typical corporate libraries are 200,000 to 1 million compounds. So that's, yes, there's a dot there. Hard to see, but there's a dot there. E-molecules, which is a commercial vendor collection company. So it actually collects sort of various vendor libraries, has something on the order of about 20 million compounds. Real, which is one of these virtual vendor libraries, actually has 2 billion compounds in its elaborated space, and actually has a lot more in its unenumerated space. And so really you're talking about the ability to screen billions of compounds, as opposed to your corporate collection where you're looking at hundreds or 1 million compounds. And that really gives you access to a lot more diversity and things like that you would not have by just screening your corporate collections. And I will say we do routinely screen billions of compounds these days. So again, cloud computing resources will enable this type of activity fairly easily these days.
So how long does it take, Amy, sorry to interrupt,
Yep.
to screen 2 billion compounds, let's say?
So that varies, of course, depending on how many compute resources you can get.
Yeah.
Something like ligand-based searching can take hours so here at Ferring, we actually bought access to Orion, which is OpenAI's platform for sort of that they've enabled within the cloud. And so you can literally do a screen in hours. So you can run multiple ligand-based screens on real in hours. You can do a structure-based screen usually in days, over 1 billion compounds. So
good, thank you.
Yep. But obviously you are spending quite a bit in terms of compute cost and obviously in industry we're actually paying for that compute cost, unlike in we don't have supercomputing grants or anything like that. So we pay for that access and for all the compute time that we use for that. So and this is just and I don't have very much time. Okay, I will skip this. This is just a retrospective analysis of sort of some lead series that came from virtual screening efforts at Janssen. Free energy methods. Okay, let me talk about this really quickly since it's probably of interest to you all. Free energy methods obviously have been around for a long time. Jorgensen, decades ago has started doing free energy with small molecules and proteins. But really for pharma and drug discovery, FEP really started about 2015, a little bit before 2015, because most of us were beta testing it by then, with Schrodinger's FEP plus, and this Jack's paper that came out, and really that hyped FEP significantly within the pharma industry. And I say hyped because there were a lot of issues with FEP at that point that I can talk about. But that being said, FEP is now being more and more routinely used in pharma and within pharma, and hit identification and hit to lead and lead optimization programs. There are a number of different additional methodologies now that are being implemented by other companies. Mo has a front end for AmberTI and a few analysis scripts. They're not doing any development on AmberTI, but they do have a front end for it. BioVia implemented Charlie Brooks's multi-site lambda dynamics. Crescit implemented a version of OpenMM's FEP. And then I have OpenEye's non-equilibrium searching method that in gray, because actually they don't technically release it until Wednesday. So, but additional software companies are getting into this space, but by far, Schrodinger is still by far the lead most used software in terms of FEP within pharma. Let's skip this. There were lots of issues in the early days of sort of how you ran FEP and sort of the caveats and pitfalls and say how things can go wrong with FEP. But there have been a lot of sort of best practices and things like that. And really we've gotten to the point where FEP is now being routinely used in some drug discovery projects. There's still a lot of drug discovery projects where the proteins of interest are not necessarily amenable to FEP calculations. And obviously, if we can consider FEP a success, Nimbus, which is a company that was in part founded by Schrodinger and really utilized their free energy platform, really drove a lot of their initial drug discovery projects, first around NASH and then a second one around TYK2 using free energy calculations. And so their entire claim to fame is the use of free energy predictions in driving their drug discovery projects. Now I will say, compound potency is not necessarily the only thing that is involved in optimizing a drug for optimizing a compound for as a drug. In fact, it's probably the easiest thing to do. Optimizing potency in conjunction with drug-like properties is actually the difficult part. And so really it's those drug-like properties that we struggle with in drug discovery projects. Okay, yep, ending remarks. So I'll say there is unprecedented interest in computational chemistry right now in pharma and biotech companies. There's been a lot of publications, interests, and things like that that have been, in the last 2 years. I would say, this was a article about in silico medicines, sort of what they claimed as a successful drug discovery project in 3 weeks. I'd say most people in pharma think they pretty much just analog known compounds, but anyways, it definitely has gotten a lot of newsworthy attention, and so there's just a lot of interest in AI methods, ML methods, in all stages of pharma. Well, let's skip that. Obviously, what came out, a couple of weeks ago, no, almost a month ago now, DeepMind's AlphaFold, and I guess in some regards also Rosetta Fold, predictions of protein structures and things like that. I say the other reason I think that there is really a crazy interest in sort of computational chemistry right now is because we all got sent home for COVID, and there was a realization, all our lab activities essentially shut down, and really we were only allowing essentially one chemist in a row of hoods, and they were there, and so you really had to prioritize your compounds for synthesis, and really that's where computational chemistry came in. We by far were super busy during COVID because every single compound got sent to us when normally they might have sent us, the chemists might have sent us, half of their compounds, and the other half, it would have been super easy to make, so they just made them. And with COVID, they couldn't just make them. And so they really were emphasizing prioritization of compounds for synthesis. And really that has held over, even though, the chemists are now fully back into labs. And lastly, I'll just say, there are a lot of opportunities, within industry for computational chemists, within biotech, within pharma. Find a job you love, find a place that most matches your interest, your ideas and what you want to do with your career. And that may not be in industry. There are plenty of people who do a postdoc in industry and find out they really don't like industry and go back to academia. So really find something that you'll love, because that really will enable you to do the best you can in your career. And with that, I think I'm done. Pretty sure. I think I'm over. Oh, network. Yes. I will just say this. Networking is very, very important. Computational chemistry in industry is very small. It is very important for you to network within computational chemistry, with academic groups and things like that. So please do that if you're interested in pharma.
Well, thank you very much, Amy. This was probably the most insightful talk I have heard about actually what job opportunities, what is happening in industry. Really nice coverage of the skills that you guys are interested and how it works. Thank you very much. I have a ton of questions, but I don't want to take too much time. So let me actually just, because I think it's important for students to find out how it works. So I have two questions. My top two questions are: how did they find you when you received the phone call from them, from Janssen? How did they spot you? And then complementing this question is, what do you look at when you're trying to hire someone? Do you look at their computational skills per se, or do you look at other things more importantly and then computational skills? What do you look at when somebody is applying for a job?
Good questions.
Yeah.
Sorry. So I can tell you, when I applied to industry for my postdoc at Janssen, I didn't know anyone at Janssen. They really picked me out because of two reasons. MD, they really wanted to have a postdoc who was doing MD, because that was a technology that they wanted to bring more experience in-house. The other one was membrane proteins. There was a specific interest in doing modeling of membrane proteins. You can see that most of my projects were membrane proteins during my postdoc, and so that was really the two things that brought that really that attracted them to my CV. Now let me find my slide. Yes, this one. Oops. I figured somebody would ask this. my brain dump.
Yeah, good.
So things we look at, when we're evaluating potential new hires. Obviously, technical ability is one. Certainly when you're coming from academia, we don't expect you to know every methodology that computational chemists within industry use, but you should demonstrate enough technical ability, within your background area for sure. And that's by far, that's expected, right? So if you lack technical ability, you will be filtered out regardless. A lot of times we're looking for a particular fit within a group. So if your background is MD and our entire group is full of MD people, that's not necessarily a great fit for a group, right? A lot of times we're looking for diversity in background talent. I specifically actually, for Sunny's position, I specifically recruited for somebody with MD background, because the other person in our computational group was actually came from a QM background. So, and had been in industry for dozens of years and really had a lot of cheminformatics and experience and things like that. And so I really was recruiting somebody who had more large protein and MD background. But that really depends on the particular group and what they're looking to fill. Larger groups may have more overlap and things like that. Communication, interpersonal skills. So I can't stress how important this is. So this communication, interpersonal skills, at all aspects when we're interviewing you. So that can be from the phone screen, which tends to be a little bit informal, where you're just sort of chit-chatting. The seminar that you give and how clear it is. Oftentimes we will invite chemists and biologists to these seminars, and we really are looking to see if you can communicate with them clearly in an efficient manner.
I see.
Our one-on-one interviews. And, I put lunch and dinner because historically we used to have in-person lunch and dinners, but obviously in sort of COVID times, everything is done on Zoom. But those lunch and dinners are a part of your interview. And so there have been instances where people have said and done things in these lunch and interviews, which say are not necessarily the most professional, and please don't do that. Publications and track record of accomplishments, publications do matter. They matter to you, not just people coming from academia. They matter, they mattered for me getting my position here at Ferring. They demonstrate a record of accomplishments. Publications, patents, and things like that. They're also the only things you can talk about when you come from industry, when you go to do an interview. You cannot talk about any of your active projects and things like that. And so, if you come from a company or a place where you don't have a lot of publications, you will have very little to talk about during your interview. You'll only be able to talk about sort of vague things, and that makes it very hard to interview. So just keep that in mind. There's actually, even within pharma, there's a strong emphasis on publication. And then lastly, pharma experience, but obviously if that really depends on the level of hire we're looking for. So, if we're looking for somebody straight from academia, then we're not expecting pharma experience, but
right.
But really a lot of sort of the let's say more soft skills. We're looking for people who have leadership potential, who are willing to learn and grow, who have enthusiasm for the position, and who are really motivated to produce results. And really this motivation for us in pharma also has a timeline, right? So pharma has very strict timelines for calculations that really, we can't take months and months to complete a calculation and stuff. And so you really do need to have the motivation to produce results and produce results quickly.
I see.
And then, potential for working with other functions, problem solving, and even past interactions. I don't know how many people really understand this, but when we go to, as someone in industry, when I go to a conference or a meeting and I talk with students, I remember them.
Sure.
I remember my dinners with Sunny when we were at Biophysical Society. There was no science discussed, but I remember that interaction with them. Those all matter, and those all come back. So when you are at these places, please be a professional at all times.
Absolutely.
Say it that way.
Yeah, cool. So again, I mean, the seminar is open to any questions, so you can either raise your hand or type your question if you are more comfortable into the Q&A or the chat box, and then we can bring them up with Amy. So this again, I think I have quite a few questions. So then, you guys have internship programs also, Amy, at Ferring?
Ferring currently does not have internship programs, and internship postdoc positions in computational chemistry groups have, let's say, depending on the group size, can be really challenging because there is a lot of time and effort dedicated to training of the interns and postdocs, and you almost need a group big enough to really support those activities. I'll say it that way. When I was at Janssen, I had a postdoc, or a group in the US was actually 18 people. So it was actually significantly large. And I could dedicate significant time to that postdoc and really training her. And actually, she joined about 6 months before COVID lockdown. I can tell you, it was very hard to get her the exposure that a normal postdoc that was on site would get, because she was not having those sort of informal interactions with other colleagues and things like that. And I spent a lot of my time really organizing sort of informal meetings with her and other functionals, groups and things like that to really get her that exposure. But those things, I'll tell you, COVID and being remote and things like that does make it a lot more challenging to get postdocs, integrated and really like the exposure that they should get.
So I see some questions in the chat box. I guess you addressed almost everything during your talk. They were posted earlier. So one person is asking what kind of tools, methods you used during your PhD, and what about the time when you joined pharma? So I guess you addressed the tools that you need to work with every day already in your talk. The other question is about cryptic binding sites and binding pockets. To what degree, how serious do you take them and you go after them? Let me put it this way. Nandan, you are asking this question. Do you want to sort of ask it yourself?
Yeah, I mean, I think, yeah, sorry, my video is not working. Already answered the question. But
Yeah,
like,
okay.
was like yeah, exactly what Imad said, like to what extent do you take the cryptic binding pocket seriously? Do you really go after them, or you like think it's not something could be useful? Is there something like that?
So so keep in mind, computational calculations are never done in isolation in industry, right? You can couple your cryptic binding prediction with mutagenesis, with potential assays, potential if you find or you identify a compound that you suspect binds in that cryptic pocket, you can harness chemistry resources to do probes of that, analogs of that compound to really determine whether those analogs fit with your model of that cryptic pocket and things like that. So there's a whole host of sort of biological things that can be done to really try to interrogate whether that cryptic pocket is of interest. And not only is it of interest as a pocket, but a functional interest, right? Does that cryptic pocket actually produce a functional effect that you're looking for in terms of your disease indication?
Moyon, you had a question about biologics.
Yes, thank you, Amy. Thank you for your talk on all of your like experience. So I just want to know like what's your biologics, especially nowadays we have antibodies protects in pharma. So yeah.
So PROTACs are generally still considered small molecules. I'll say it that way. Biologics tend to be more antibodies and protein engineering. Antibodies are often done in industry right now, and computational chemistry is involved in antibody design, although for the most part, antibody design these days is done using phage display, and it's very fast and an experimental method, and they can get potent antibodies without necessarily the need for computational calculations. I'll say it that way. Where computational calculations come into play oftentimes is when they're looking for bispecific antibodies. So antibody that will bind two different things. And so they're able to sort of optimize for one and optimize for the other, but then they try to optimize for both. And a lot of machine learning algorithms are actually used in sort of trying to predict the one that will bind to both the best. Where computational chemistry or computational methodologies come into play a lot more for antibodies is actually in the development phase, where you're looking at formulating your antibodies for production. So things like stability of your antibody. So loop movements and things like that could greatly impact the stability of your antibody. If there are large hydrophobic patches of your antibodies and things like that, they could result in aggregation. So really, computational calculations within sort of the large molecule sphere tend to be, I would say, there's a lot more in the development phase of things.
Nice. Okay, great.
Yeah,
so let me ask you the next question, Amy. So what was, if there was anything, like what was the most difficult aspect of transitioning into industry for you from academia? Or maybe there was nothing like that, so you just made a smooth transition. But did you notice anything, a major change that you had to make in your life, in your style, in your science? When you moved
I think it had a lot to do with how you think about projects, how you think about moving projects forward. You're no longer thinking about the science of a particular target,
I see,
the biological function of a particular target. You really
right.
are focused on finding the right methodology to push a project forward. And by pushing a project forward, I mean designing that drug to be a clinical candidate. I mean, that is your end goal. And so that it requires a different thinking process. And I can tell you, every computational chemist that comes to industry from academia has a huge learning curve.
Just for that.
There's a huge learning curve just for that. There's a huge learning curve in terms of methodologies you use.
Yeah.
There's a huge learning curve on how you interact within your project teams and things like that, and how you present your data and things like that. So and learning the drug discovery process on its own. Most of us are not exposed to drug discovery, in academia, and certainly not industrial drug discovery processes. And so it's a learning curve.
Absolutely. Okay, sure. Jale, you can ask the last question, maybe. Go ahead.
I have a quick question about the pathway. Like, what does this path looks like 5 years, 10 years down the line? Because in academia, we know that, for example, 10 years, you probably are a full professor if you start your assistant professorship. But how does it look in industry?
In some regards, it really depends on your interest. So, we have, at Janssen, we had people in the group who had been there for 20 years, and they were very experienced computational chemists, but really what they wanted to focus on was leading and supporting projects and drug discovery projects. There are other people like myself who really wanted to lead a group. And so that's where my career went. I can tell you, my manager at Janssen is my manager now at Ferring, and she actually leads a lead generation of pharmacology group, which is no longer just computational chemistry. It includes in vivo and vitro pharmacology, screening, biophysics, essentially all modality agnostic functionalities within lead generation. And so, you can move up the pharma chain if you want, if that's your interest. It really depends on your interest within pharma. So
interesting. Thank you.
Yep.
Definitely, you can ask the last question if you're a student.
Hi. Okay.
Yeah.
Yes. Hi, Amy.
Hey.
I'm wondering if we can apply for other positions other than computational chemists as graduates of, theoretical biophysicists.
So yes, you can. In fact, let me go to slide.
Another slide.
That's great.
There you go. So computational chemistry, yes. Computational biology, CAD, computer-aided drug design, computer-aided drug discovery, molecular modeling. There's a whole host of sort of names for groups, and titles. So I can't really. Sometimes you're called formulation. Sometimes you're called when you're searching for a position in industry, you can search outside of sort of computational chemistry as a title for your job search and things like that. And really don't be focused on sort of the exact title of what pharma usually has titles along the lines of scientists, senior scientists, principal scientists. Those titles vary by company, and so what is a scientist in one company is not a scientist in another company, and so you do really need to look at the job description to see if you meet the needs of that particular position. Do not be afraid to apply for positions that you don't necessarily meet all the qualifications. I can't stress this enough. You could see in my career when I was at DART, and unless my former manager had reached out to me, I would never applied for that Janssen position because I did not meet the job requirements. Now, to be fair, I was perfectly happy at my job at DART, but be open to applying for positions that you do not necessarily meet 100% of the job requirements. Now, I will say, usually the one job requirement that you do need to at least somewhat meet is that industry requirement. If they are really looking for someone with industry experience and you have none, that probably is going to be a disqualifier.
Yes.
But everything else in terms of methodologies and things like that are usually kind of flexible. We usually just put a wall, I'll say, a list of just things that people could be who have backgrounds in could be, amenable to our group. So.
Okay, well, it's already 4:20. Thank you very much again, Amy. This was really useful. I mean, it helped me a lot. I'm sure the students also enjoyed it. I look forward to more interaction with you and your group in Ferring and more opportunities for the students. Thank you so much, Amy.